Welcome to my PowerPoint presentation on the genomic analysis of Avibacterium paragallinarum isolates from an infectious coryza outbreak in the U.S. in 2019. Infectious coryza is an acute avian respiratory disease. The symptoms include decreased feed and water consumption, decreased egg production, swollen heads, and increased nasal discharge. The disease has significant economic impact in layer operations, causing significant drop in egg production and poor growth performance. It also has an impact on condemnation rate increase in the broiler industry. Infectious coryza is caused by the bacterium Avibacterium paragallinarum. This is a catalase negative, oxidase positive, short gram-negative rod. This organism is fastidious in that it requires NAD for growth. Some regions of the world report NAD-independent strains. On blood agar, A. paragallinarum colonies are pinpoint, non-hemolytic, smooth, circular with entire margins, and will satellite around a Staphylococcus nurse streak, which is shown here in the picture on the slide. SIVA Scientific Support and Investigation Unit has received 9 clinical cases during the 2019 outbreak of infectious coryza in the northeastern United States. Cases presented with infectious coryza symptoms mentioned prior and consisted of 132 whole heads from 24 layer houses aged 15 to 92 weeks from 4 separate states: Delaware, Maryland, Pennsylvania, and Virginia. These samples typically yield mixed cultures of other significant respiratory pathogens such as ORT, Galibacterium, Erysipelotheryx, Enterococci, and E. coli. Upon receipt of the head samples, the beaks were removed for nasal swab collection. All samples were plated for isolation on 5% blood agar with a staph nurse streak and also on chocolate agar plates. Cultures were incubated for up to 3 days, and isolates confirmed as Avibacterium paragallinarum were stored for further analysis. Bacterial cultures were identified using the NHAPI identification system. Historically, differentiation of Avibacterium paragallinarum isolates has been by serotyping. Page first proposed 3 serotypes, A, B, and C. Hume decided there were 8 serovars, naming them HA1 through HA8. Blackall modified this method by joining both Page and Hume typing schemes, and now this is the widely accepted method of serotyping. This classical serotyping method types Avibacterium into 9 serotypes: types A 1 through 4, B 1, and types C 1 through C4. However, classical serotyping is difficult and reagents are hard to acquire. There's been an attempt to correlate serotyping of Avibacterium paragallinarum with PCR amplification of the hemagglutinin HMTP210 hypervariable region. This was done by Sakamoto et al. In 2012. Similarly, in this study, we trimmed the region 2 of the HMTP210 gene, utilizing the primer set 5:1 shown here. This yielded roughly a 1600 base pair product, and we created a phylogenetic tree in an attempt to find a correlation between serotype and the HMTP210 sequence. Reference sequences obtained from the National Center for Biotechnical Information, or NCBI, of known serotypes used by Wang et al. In 2016 were included in this analysis. We are relying on the accuracy of the traditional serotyping method for these reference sequences. This figure shows the hemagglutinin gene HMTP210 hypervariable region tree. The reference sequences are in black, and the outbreak strains are color-coded by state. This analysis could not clearly separate all the reference sequences with known serotypes accordingly. Serotype A was separated into 2 major clusters with some minor outliers. Serotype B has 1 grouping, and serotype C shows 1 major cluster with other C references mixed with other serotype A references. However, the isolates from this study all clustered with the majority of serotype C. To further attempt to characterize the outbreak isolates, we performed a pangenomic analysis. All genes present in all strains were assessed and divided into core and accessory genes. Core genes are those present in greater than 99% of all genomes sampled and are usually required for function. The rest of the genes were classified as accessory genes, which were not present in every strain and usually not required for function. A total of 46 outbreak strains were sequenced using the ion torrent S5. In all 46, there were 4,284 genes present. Of these, 1,396 were classified as core genes being present in all 46 isolates. The core genes were concatenated and assessed for single nucleotide polymorphisms. The pangenomic tree was built from the maximum likelihood alignment of all 1,396 core genes. This slide shows the pangenomic tree of the core genes of the outbreak strains. The relationship among core genes shows that isolates from Virginia are in a different clade than those from the other states of Delaware, Maryland, and Pennsylvania. There are a number of methods used to analyze a group of isolates with the goal of understanding their epidemiology, pathogenicity, and diversity. Phenotypes can distinguish isolates biochemically, by serotype, or simply by morphology. Molecular PCR methods can detect the presence of specific virulence genes, further characterizing an isolate. Multi-locus sequencing typing, looking at a small number of specific genes and SNP differences between them. Whole genome sequencing can provide additional information to aid in understanding diversity among isolates. In this study, we focused on the analysis of the hemagglutinin gene for serotyping Avibacterium paragallinarum. This group of isolates gave an indication of its association with serotype C. While this method may be useful in determining serotypes A, B, or C, caution should be taken as some serotypes do not cluster distinctly. Complete serotyping to the level of Q methodology is still elusive, justifying further research in molecular serotyping following the footsteps of Sakamoto in 2012, Wang in 2016, and Araya Hidalgo in 2017. Instead of looking at a limited number of virulence factors, the second focus of this study was the pangenomic analysis of those 1,396 shared core genes amongst the outbreak strains. It suggests distinction among isolates from Virginia. What is the common denominator between the 3 states of Delaware, Maryland, and Pennsylvania? Why is Virginia different? Is it simply geography? We're expanding our research on infectious coryza, gathering more isolates and more information, looking beyond New England into the rest of the United States and the rest of the world. Thank you very much.